Hi everyone. Just gonna share my screen. Hang on one second. Okay. Just gonna give everyone one more minute. All right, let's get started. All right, hi everyone. My name is Lisa, and I'll be talking today about downloading sequence data and quality control. Just a little bit about me. I'm a first-year PhD student in the Systems and Computational Biomedical Sciences program. Yeah, I think that's it, SCBM. I'm currently rotating with Dr. David Fenyo and Dr. Teresa Devoli. My interests are in cancer, proteogenomics, and precision medicine. So let's get started. So today we're talking about downloading publicly available data. Where can we find it? There's a lot of different places that you can find publicly available data. One place that we'll be talking about today is the Sequence Read Archive, which is SRA. It's an NCBI run consortium, I guess. It's a repository for a bunch of different sequencing data. It's huge, it's very commonly used. Another place that you can go is the Gene Expression Omnibus, which is GEO, or the European Nucleotide Archive, ENA. There's a lot of different places where you can find publicly available data. The NYU Core has a bunch of data. There's a lot of other consortiums that have data like TCGA, CPTAC. But today we're going to focus on SRA. And basically it's just a large repository for sequencing data that's publicly available. The main mission of the SRA was to make sequencing data available to the research community, to enhance reproducibility and allow for new discoveries. And I'm actually going to exit here. And we can go onto the SRA website and do a quick search. So if you go to the NCBI SRA website here, let's just say we want to look at, I don't know, cancer sequences or something. Here we go. So then a bunch of things pop up, a lot of accession numbers. We can narrow things down, like we only want human reads. If it's gonna ever load. There we go. We can narrow things down, like we just want RNA, single versus paired end data, things like that. So you can play around with this and look up different types of data sets. And download them there, and I'll go into a demo on how we can download them via BigPurple or any other high computing cluster. I'm not gonna go into presentation mode because I'll be flipping in and out of the PowerPoint, but hopefully everyone can see this. So one of the first steps that you should always take when you're looking at sequencing data is quality control. So why do we want to quality control? We want to see if our sequencing data is ready for alignment. First, we want to check that our adapter sequences are removed and we're getting rid of low quality base calls. So if you remember from Dan's lecture a couple weeks ago, I believe, so this is, for example, paired end data. And if your sequences are still containing these adapter regions that are kind of towards the ends of the reads, or if you have some sort of low quality base calls, you're going to want to get rid of those before you start aligning. But today we're not going to focus on the trimming part. We're just going to talk about how to get there, how to see our sequencing data, just QCing it. And probably one of the most commonly used tools for this is called FastQC. I'll go into how to use that after I demo how to download the data. And essentially the general workflow that you'll usually go through when you obtain some sort of sequencing data is you download data, you QC it, you'd probably have to trim right after you QC, QC again to make sure that your trimming step was successful, and then align, and then you do some sort of analysis. And today we're gonna mainly focus on downloading and QCing. So if we open our terminal, let me just make sure, sorry. Here we go. If we go into our terminal, or for anyone on a Windows computer, you can go to PuTTY. So we're gonna wanna SSH into BigPurple. Just a little thing that I did there, I pressed the up arrow on my keyboard because the last thing that I used in this terminal was SSHing, and it pulls it up. So that was like a little quick step. Enter your password. Also just make sure that your big IP Edge F5 program is connected and that you're VPN'd into the NYU VPN. So just make sure you're connected. I believe Angelina went over that last week. Sorry, I can't see the comments right now when I'm sharing my screen, so I hope there's nothing going on in the chat right now. If there is, hopefully the TAs are taking care of it. Yes, okay, so we're in BigPurple. So we're gonna go to our class directory. So to change directories, we type CD, and we're gonna go GPFS, data. I'm clicking the tab key when I type in a couple letters, and it usually fills it in if it's able to recognize where I'm going. That's why it sometimes fills in right away if that's like, if you didn't know what I was doing there. Yeah, so data, and then we're going to courses, BI20. So this is our class directory. You can do ls to see what's in it, or I usually use the command ll, which is just long list, I believe. And that kind of gives you some more information. Here, let me make this a little bigger. Yeah, so LL kind of gives you some more information. These are the permissions allowed, and I'll go a little bit more into that later on. And you can kind of see, this is, I believe, the folder or file size is over here. When it was created, I believe, or the last time that it was used. If you do LL-H, that's human readable, so it'll tell you even more details, like it's a, yeah, sorry. Okay, so I already started working on some stuff for downloading data because it takes a bit of time. So I'm just going to show you what I've done so far, but we can go through this again and I'll go along each step with you. If you go into the Lecture 13 Download QC folder, and we look at what's in there, I have an example file there that is kind of an example script for how we can download data. And we're using the SRA toolkit, which I believe Angelina showed you guys last week. If you look at module avail, SRA, here's the SRA toolkit, and that's what we're gonna use to obtain the data from SRA into our BigPurple environment. So, if you want to take this file that I created, which we can also open, nano download. Here's like an example script that I have. It's like a bash script, right? So we have our sbatch commands here. This is kind of how every shell script should start, which is the .sh here that specifies that it's a shell script. Oops, just double check. Okay, I'm seeing that someone can't access the data. Interesting. Okay, hopefully you can get into this folder. It should be available to everyone. Yeah, make sure that you have your VPN running so you can SSH into BigPurple. And then if you're trying to get into the class directory, it is only on BigPurple. If you're working with prints, it will not be in there. We're trying to get a folder available for us on prints, but I don't think we have that yet. Let me just make sure. Okay, yeah, so if you're working with prints, it's unfortunately not on there yet. If you do open the slides, they're on the course website. The slide that I have here, it should be slide 7, will have the example script, so you can copy that into a new text file in your home personal directory on prints, sorry. So you'll be able to run all of these commands, just you won't be able to copy my personal file that I made in our class directory on BigPurple. Okay, so if we're looking at this sample script, we have the job name that I have here, so that's the download_example. You can give it any name. Mailing, I'm telling it to email me anytime the script ends or fails. So if it fails, it'll send me an email saying your job failed. When it ends successfully, it'll also email me. It won't email me for these other things here that I've just added a comment for. You can tell it, if the script just begins, the job begins, you can tell it to email you, or just specify all, but here I'm just telling it end or fail. For the mail user, you put in your email here. For me, it's my KID at NYU Langone, but it could be whatever email. I'm giving it 1 CPU to run on, 8 GB of memory, 2 hours of time, and then for this output, this is an output, this is the output directory for my log file. So when you submit a job, it will create a log file kind of telling you what is going on in your script, if there's different messages coming out of it, and you can specify where that's going to go. And then the last thing is the partition. So I'm telling it to go to CPU short. I'm loading 2 modules here. I'm loading the SRA toolkit, and I'm loading fastqc. So the first thing that we're going to do is fastq dump. And that's essentially telling the computer to get these files that I'm specifying, which is this accession number, which I'll go into in a second. I'm telling it to go to this accession number in SRA. Split files is a flag that I'm using here, which is going to separate the forward and reverse strand because it is a paired end read. I'm telling it to zip the file here. Then this -o is telling it the output directory that I'm going to go to. So here I'm telling it to go to this folder that I named downloads. And I'm also giving it this flag orig format, which is, I believe, just keeping the header, the original format, in the fastq file. And then I'm going to run fastqc on my new files that I make from downloading. And this is a pretty easy command. There are other flags that you can use, but for now, I'm just telling it fastqc, and then the output is this qc folder that I made, this directory. And then I'm telling it the first file, which after the download, it creates these 2 fastq files that are zipped over here. Essentially the same accession number, except _1 for the first read and _2 for the second read. That's the forward and reverse. So I'm going to exit out of Nano for now. And if you want to copy that file to your home directory, you're going to go back. Let's go back home. So if you do cd tilde, that'll take you to your home directory. So if I go back, these are just my own personal files that I have. If I want to copy that file, I'm going to do cp, give it the path to that file. So that was gpfs, data, courses, DI20, lecture 13, download, and then download _example.sh. And then make sure you do a space here. I'm going to say dot here because I'm telling it to essentially drop that file, that copy, into the directory that I'm already in, my working directory, which is my home directory. So now let's check again. And that file should be here, yes, download example.sh. Okay. So hopefully everyone is able to do that. Let me just check the chat. So is everyone able to, oh, sorry. If you want to exit Nano, you do Control X. Okay, hopefully everyone was able to get this far. You can also follow along in the slides that are available on the website. The commands are pretty much all there. Okay. So hopefully everyone was able to copy that into their home directory. Again, this is just in the slideshow, there's the example.sh is all here. So you should be able to copy it into wherever you're working from. Okay. So again, you can nano back into this file. Sorry, I'm using Nano. You can use Vim or whichever text editor that you feel comfortable with. I'm just used to Nano. So if you actually want to run this, I believe people have already been changing things in this file. Please don't change anything in the file that's in the lecture directory. So copy it over to your home directory and then change it. So for example, I'm going to put my email here. I'm going to tell it this is the output or whatever. And before, actually, so I'm going to change, I'm going to get out of Nano for a second. So Control X. I did modify something in this file, so it's going to say save modified buffer. I'm going to say Y for yes, and then just enter. And I'm going to make some new folders. So I like to keep my files pretty organized, except for this home screen, which I do have a lot of files here. But if we want to make new folders for, we want to make new folders for where our outputs are going, you can type in mkdir, and I'm going to make 3 new folders or directories that are called log files, downloads, and QC. So this should have made 3 new folders, downloads, log files, where is it? And QC. And I'm going to specify in my download script where to put all of those things. So for example, if I want my output log file to go to log files, I'm just going to specify that here, right? This is just like a normal output, and I'm not putting anything before this because I'm starting it from my working directory where I am right now. So I'm not going to put anything before that. I'm just going to specify that here, that this is going into the directory that I just made called log files. And then I can name this output log underscore %j. The %j is going to take the job number that it creates when I submit this as a job. You don't have to keep that there if you don't want to. I like to have it there just because when you start running a bunch of jobs, it'll be easier to get a little bit more track of them. Then I can specify here that I'm outputting my FASTQ files into this directory called downloads. Again, I'm not putting anything before. I could write here GPFS, like the full path, GPFS, I don't know, home, and then my Kerberos ID or something. This would also work. This would go to the same place, but I don't have to do that because I'm already in my home directory and I don't have to specify that whole long path, basically. And then again, here, I'm going to be calling these new files that the download creates by calling it from this downloads directory. This is what the file name is going to be. And then I'm also telling it to output into this folder that I made, which is called QC. So if we exit out of here, Let me just check the chat. Okay, hopefully everyone is able to get this far. Okay, so permissions. So permissions are really important in BigPurple and pretty much any high computing cluster. These little letters over here, when you type in LL, will show you the permissions. And there's actually a pretty useful link that I think, yeah, I put it in the slides, but it opens here. Chmod is the command that you can use to change the permissions for any kind of files or folders or directories or whatever in BigPurple. The different letters here, RW, and I believe X, yeah, they stand for read, write, and execute. So and then the positions of those letters is also very important. So the first one is for the owner, so that's you, that's if it's your file. Then it's for the group if you're sharing it with people, and then others, just anyone else in that environment. 777 is a common, hold on, where is it? Is a common, I guess, command that you can use after chmod to kind of allow basically anyone to read, write, or execute your file or directory. If it's important information or like secure data, you probably wouldn't want to use 777, just because it gives literally all of the permissions to anyone looking at that file. So you can use these different number codes for other permissions. But for now, I'm just going to do chmod, oops, 777 for my download file here. And now it'll turn green because it's now basically, and now the codes here have changed, because now it's basically accessible to essentially anyone, I believe. And then I can also do that to my folders that I've made. So if I do chmod 777 to downloads, now that's also changed here. And I can do it to log files and that QC file. So now all of these should be available for me and anyone else pretty much to change anything in it. You can also do, like, if I wanted to just do it for me, I believe I could just do chmod 700. Let's say download example here. So now that has changed here. It's pretty much only accessible to me. If you look right here at the RWX, there's nothing available for group or others. Okay. Sorry, I'm just looking at the chat real quick. If you can't open the lecture slides, I'm not really sure why that is, because it's a Google Slides. Sorry about that. Hopefully we'll get like you can maybe have like a PDF available or something. Okay. Yeah, so this is essentially ready for running. I'm gonna say maybe don't run this right now on your computer, because I think BigPurple might be a little backed up right now. And if we all submit these jobs, it definitely won't run for everyone. I was even having some issues getting a job to go yesterday and earlier today. So just for your own knowledge, if you were to submit this job, because this script is technically ready for you to run, you would just do sbatch and then this downloads example, and then it would run. So I'll run it here. I can do sq, my Kerberos ID, and we see that it's running here. I'm just gonna do scancel because I don't really need this to run right now. If I do scancel and then this job ID, it'll cancel that job that I just started. And now I see sq, my Kerberos ID, and it's gone. So I canceled that because I've already run this in our lecture directory. Something I should have mentioned before. So I'm going to show you exactly which data we're going to get, which I realized that I haven't gone into yet. So if I go back into this file, this is the accession number that I'm getting from SRA. If I copy this into the website, you can see exactly which experiment it came from. So this was run on an Illumina high-seq machine. And these are, I believe, fibroblasts that have been infected with the HSV1 virus. I believe it's herpes virus. You can actually see like an abstract of where this data is coming from. It'll tell you a lot of other information. So this is RNA sequencing data coming from cDNA. It's paired end data. Something to just look out for when you're looking for data. This is the SRX number, which is, I think, just linking you to like the whole project. What you want to give SRA when you do FastQ dump is the SRR number over here. That's the actual run that you want to give it. So sometimes in the SRX numbers, you'll have multiple SRR numbers, and you want to just run those separately. This one only has one SRR number here, so we don't have to worry about that. But I did tell it this number, so always just look up the data that you're going to be downloading, right? So this is important that we know that these are human cells that have been they've been infected with virus, so there's going to be some sort of something interesting happening in the sequencing data, right? It's not going to be perfectly aligned to just human genome, right? There's going to be some viral DNA or RNA in there. Okay, let's see what's happening in the chat. Yeah, okay, sorry. I know that a few people are using prints. There is unfortunately, this is not available on prints yet. I'm really sorry about that. We're trying to get it on there. I just don't have a prints account yet. So if you are working on prints, unfortunately, these files are not on there, but if you look at the slides, you should be able to get the commands and everything in the script. Okay. So I already ran this before, so I'm not gonna rerun it now. I did cancel my job. If I go back to our class folder, I can show you kind of what the outputs look like. What is it? Data, courses. Okay. Let's see. Okay. If you are doing stuff in the class directory, it will save here. So there's different stuff here now. Please don't. If you're going to change stuff, take it into your home directory. So hopefully everything stayed the same here. If I go into my downloads folder, these are the files that come out after the download. They're zipped files. I'm not going to open these. They're gigantic. But this is essentially the sequencing file. So this one over here is the first read. This 2 is the second. That's the forward and reverse strands because it's paired end data. I'll go back one folder, one directory, and if I look at my log files, I should have one log here. There was an error happening before. I think it was because BigPurple kept canceling my job and restarting it, and it really confused the program, but it downloaded fine. If I had these on a normal basis, I would have restarted this probably if this had occurred for some kind of important data. But because this is just a demo, I didn't want to have to redo the whole script. But after it downloaded, it ran FastQ over here. So this is in my log file again. Just press Q to exit that. I said less and then the file name. So Q to get out of that. And then I can go back to my QC folder that I have here. So FastQC outputs HTMLs and then this zipped folder with all this other information that it provides. You don't really need to look into this folder. This has like kind of everything step by step that it's generating. But the important thing here is these HTML files. So those are going to tell you all the information from your QC run. I go back here, let's see. Yeah, so I went over these flags before. Yes, so QC, we're gonna wanna copy those QC outputs into our local computer. So you can do this using SCP and then your BigPurple account, like your KID at BigPurple, and then the file path, and then the path on your local computer. There's also another way to copy these files, which is using a program called FileZilla. And I can open it here. It's just a program that essentially connects you to the BigPurple server, and it looks like this. So this is something that you can download. It's free. If you wanted to, so I already have my BigPurple account ready here, but if you wanted to do a new server to connect to BigPurple, and I believe it's the same process for prints, you would just do, you have to tell it the protocol is SSH file transfer protocol. The host is your KID, at BigPurple. Sorry, one second. I'm just gonna, sorry about that. It's not your KID at BigPurple, it's just the BigPurple domain. Yes. The host is bigpurple.nyumc.org. I'm going to write my KID here. I want to tell it to ask for my password, and I'm going to tell it to connect. It's going to ask for my password here. So now it's connecting. So now I'm connected to my BigPurple account here. This is my home directory. I can go to the class directory. So if I go to GPFS data, courses, that'll take me to the directory for our course. And here's all my folders again. So I want to go to QC, and I don't know, let's go to my desktop here. This is my local computer here. If I open my QC folder, I see these two HTMLs here, and I'm just going to drag them over here. It's transferring those files. I think I had them there already. That's fine. Okay, so it transferred over to the files. Then I can open these files here. And they're going to open in a web browser. Okay. Just going to check the chat real quick. Hope everyone's okay. Sorry if I'm going too fast. I can slow down a little. Okay, so this is what the output looks like for FastQC. It's the FastQC report. If we go to their FastQC documentation page, this is what it looks like. I believe the link to it is here in the slides. Yeah. Maybe not. All right, I'll put it in there. Okay. So this is their documentation page. If we look at what their good Illumina data QC file would look like, all of these will have check marks here. These are kind of all of the things that FastQC is looking for. So for example, this is my file that I just ran on the raw data, right, for our FastQC. So the basic statistics look fine. The main thing to look at here is sequences flagged as poor quality are 0, so that's good. The base sequence quality is looking okay. We're kind of in the green here. These are the, I believe this is the Fred scores, and this is the precision in the read. If you look at the FastQC, like good Illumina file, it'll all mostly be closer to here, the top, like 38. Our file has some deviation, but it's pretty much okay. Again, this is the raw file. We haven't done any kind of trimming, so we'll expect to see some poor quality reads. The per tile sequence quality, I believe this is also telling us that there are some regions of poor base calls or just poor quality reads. The per sequence quality scores looks okay. But here we have this per base sequence content. So if we, again, compare it to the, sorry. Okay. If we compare it to the good sequencing FastQ from FastQC, all of these are kind of generally in the same area. They're all kind of straight lines for the per base sequence content. Ours is a little kind of all over the place in the beginning, so that's just something to look out for. The GC content, this is really important. So the GC content here, because these are cells that were infected with a virus, we're gonna expect the GC content to be a little weird. And I think we see that here, that there's this huge spike kind of higher than 50%. That should be viral RNA or viral DNA, I'm not entirely sure. I believe the human GC content is a tad lower. So because FastQC doesn't know that this data has viral DNA in it, it's gonna give us a flag here. But because we know what we're working with here, this shouldn't be that big of a deal. But yeah, if you were just working with purely human reads, this might be a little concerning. So we can keep scrolling down in this FastQC report. Let's see if there's anything else that it's interesting. The sequence duplication levels. So if we compare it to what they say should be good, should kind of look like this, where it levels off there. Ours has like a little bit of something interesting happening over here, some duplications maybe. Again, we'll probably get rid of that during the trimming process. Overrepresented sequences. Okay, so this is usually looking at common adapters that Illumina uses. So for example, we got a hit here for the TrueSeq adapter, and that will eventually get taken out once we trim the data. And then no other adapter sequences were found, so that's good. So yeah, so that was one of our files. This is the other one. This is read 2. Should be pretty similar. Yeah. So that's kind of what the FastQC report looks like. I'm trying to think if there's anything else that's important for that. I don't think so. If you wanted to do the secure copy, we can do that here. So you would have to put in, so you'd have to get into a new terminal. So for example, the terminal that I was in is logged into BigPurple. If you create a new tab, I just pressed Command T on a Mac, but if you're using PuTTY, I'm not entirely sure if you can do the same thing there. But now I'm out of BigPurple. This is my local computer that I'm in, right? This is just my home local directory on my laptop. You'd have to come back here to do that secure copy that I'm showing here. So for example, if I put in my KID there, and then the path to our file, what is it called? Sorry. Okay. Go into the QC folder, and then if I just want to grab one of these files, let's do this one. And then the path on my local computer, I'm just gonna Oops. We'll get this path here. I'll just do it there. Okay, so this should be the full command. SCP, my Kerberos ID at BigPurple, colon, the path to where the file is, and then the path, the file name, space, and then wherever I want to put it in my local directory. I'm going to put it in my desktop. So then enter, it should ask me for my password. Okay, so that should have worked. If I go to my desktop in my home directory, it should be there, plus other things. And there it is. But I would definitely recommend getting FileZilla. I know there's other applications too. I just haven't used any other ones, but FileZilla is really helpful if you want to transfer or upload things from and to BigPurple from your local computer. It's super easy. Like, you can just see everything here instead of having to write SCP and then all the paths every time. Okay. Just gonna check the chat real quick. I know, sorry, I definitely went a little too fast, but I hope everyone was able to follow. Again, the slides should be on the website. You should be able to follow everything from the slides. Okay. So for FileZilla, I don't believe you need to specify the port. So if you download it, you go to this icon here that are these little servers. This should pretty much be the only thing that you need to type in. It's the host. I don't think you need a port. At least I didn't. You also definitely need to be logged into the VPN when you do this. Just make sure you also change the protocol to SFTP-SSH, File Transfer Protocol. And then all you need is your user ID, which should be your Kerberos ID. For prints, this should be very similar, except this is obviously going to be a different host name. And then you press connect, and it's going to ask you for your password. And then it'll look something like this. So I think we're just going to end a little early today. I think I can stick around and answer any questions. Next lecture is on Thursday, and I'm going to be talking about actually trimming sequences and post-trimming QC, and what to look out for, which types of programs you can use for trimming your raw data that can be ready for alignment. So with that, if there's any questions, please feel free to ask them in the chat. I'll stick around. Yeah, the lecture slides, I can actually go to them here. If we go to this website, hold on. Oh, it's lecture 12. I don't know why it's under lecture 12. Okay, that was my bad. Yeah, the slide should be right here. So the data you can actually download yourself if you're using prints. I'm not sure we'll have these folders ready for prints by Thursday. I'm trying my hardest. But you should be able to run that script, that .sh shell script, to download the data yourself. But hopefully we will have a folder available on prints. So sorry about that. If you're trying to copy over, okay, you guys might not have permissions to copy over these files. Let me check. Yeah, so I believe you actually might not be able to, let me change the permissions for these right now. Okay, you should be able to copy those over now. Sorry about that. This is you're gonna see this is gonna be super annoying. Every time you create a file, you're gonna have to do something to the permissions. Also, if you want to change the permissions within a folder for everything, you can use this chmod -r, capital R command here, and it'll change the permissions for everything within that folder, within that directory. The R just stands for recursive. So everything in here now is available for you. Let's see. Yeah, there's other things that you can use for QC. This is just kind of the most common for this kind of data. I actually have never used anything else, but FastQC is kind of the go-to QC tool. So that's a good question. You don't need to load a library of adapter definitions for this particular situation because it's an Illumina data set, and I believe FastQC already has all of those for Illumina. If you were using maybe like a different set of adapters or a different protocol or a different platform, you might need to actually specify that. I'm not entirely sure because I haven't done that. I can go to the website for FastQC. Where is it? No, maybe I can just look here at the commands for it. Yeah, so, okay. So when I typed in, so I loaded the FastQC module and I typed in --help. That gives us the descriptions for all the flags that this command will take. There is a flag for adapters, so you can specify adapter sequences. I've never actually used this myself, but you can do that. So I'm guessing like if you're not using Illumina data, you might have to specify that. But yeah, there's a lot of different things you can add here. There's no other questions. I guess we can end a couple minutes early today. Thank you so much for joining us, and I'll see you guys on Thursday.